Dean, Faculty of Medicine, ladies and gentlemen. The topic of my presentation is in vivo correlate of protection against Plasmodium vivax Duffy binding protein in Sri Lanka. Actually, this project based on analysis of the immune response against one of the leading vaccine candidate antigen against malaria. And I want to tell that the data I'm going to present here today is a small component of a large project that was conducted at the University of Colombo to characterize the immune response against this vaccine candidate antigen. We all know malaria is the most deleterious parasitic disease in the world. Here I am presenting some of the latest data from the WHO, 2010. And of the 4 malaria parasites that attack human, Plasmodium vivax is the most widely distributed parasite. We are responsible for around 25 to 40% annual cases worldwide. And in Sri Lanka, around 70 to 80% of the malaria cases reported were due to vivax malaria. And the disease is basically confined to the dry zone of the country. The epidemiological settings of the malaria in Sri Lanka is described as unstable with low intensity. Recently, the number of malaria cases reported in Sri Lanka has dramatically dropped. You can see the drop. This is 2010, only 684 Plasmodium vivax malaria cases reported. So due to this dramatically drop, WHO recently categorized Sri Lanka as a country entered to malaria pre-elimination stage. In last 2 years, the most number of cases reported were from Vaunia, Kilinochchi and Mena, and from Monragala and Hambantota district. The most affected group of the people of basically in these 3 areas were security force personnel and the construction workers. And in these 2 areas, it was agricultural workers. And importantly, in last 5 years, more than 90% of the cases reported in Sri Lanka were due to Plasmodium vivax. The current control measures against malaria becoming less or not effective due to the emergence of the parasites that develop resistance to the malaria drugs and the vector mosquitoes that develop resistance to the insecticides. Therefore, vaccine against malaria is considered to be the most cost-effective and most effective intervention strategy. Basic of the vaccination is, one of the basic of the vaccination is the identification of the molecule that is to be used as a vaccine candidate antigen. A molecule to be selected as a good vaccine candidate antigen, that molecule should be an essential one for the parasite to complete its life cycle, one thing. And the other thing is that molecule should be able to induce acquired protective immune response. So there are 2 basic requirements. One is that molecule should be an important one for the parasite, and then it should be able to induce protective immune response. And here's a schematic diagram of the malaria life cycle. There are 4 basic stages, and according to these basic different stages, 4 different types of vaccines are being developed various parts in the world. And we are concentrating on the asexual blood stage vaccines. This asexual blood stage of the life cycle of malaria parasite is responsible for all the pathological symptoms associated with the disease. The cyclic invasion of the Plasmodium merozoite into the human red blood cells initiates and maintains the malarial infection. 4 basic steps are involved in this invasion process. And each of these steps associated or mediated by the specific receptor ligand interactions which is occurred between the parasite, the merozoite stage, and the proteins found on the human red blood cells. This junction formation or tight binding formation during the invasion process is mediated by the parasite protein called Duffy binding protein, and the red blood cell receptor involved in this process is Duffy antigen receptor for the chemokine. So you can see the myristoid and the myristoid protein, Plasmodium vivax Duffy binding protein. It goes and binds with the Duffy antigen receptor on the human red blood cells during the tight junction formation. This interaction between the Duffy binding protein and the Duffy antigen receptor on the human RBC is essential for maintaining the blood stage of the malaria parasite. And again, past research has been confirmed that naturally acquired antibodies against this Plasmodium vivax Duffy binding protein can protect or can block the interaction between the myristoid and the human red blood cell. Therefore, this Duffy binding protein is a molecule that is essential for the myristoids, and it can induce naturally acquired immune response, protective immune response. Because of that, this Plasmodium vivax Duffy binding protein considered as a prime vaccine candidate antigen against blood stage vivax malaria. The other thing is the level or outcome of the naturally acquired protective immune response developed can be influenced or can be changed by several factors, one from the parasite factors and from the host factors. During this presentation, I am concentrating on these host factors. Some of the host factors that can influence the natural immune responses are age of the patient, their previous exposure to the infection, then their genetic makeup and several other factors. So having this background, the objective of this presentation is exploring the naturally existing associations between the Plasmodium vivax antibody responses with different host factors in order to establish in vivo correlate of protection against blood stage of the vivax malaria. So we selected microscopically confirmed acute vivax malaria patients from 3 different areas, Anuradhapura, Kataragama and Colombo. Anuradhapura and Kataragama was considered as the malaria endemic areas in Sri Lanka. Colombo is an non-endemic area where the patients has acquired the disease by traveling various parts of the country having malaria. Previously validated questionnaire used by the malaria research unit of the medical faculty, University of Colombo, was used to collect the relevant data or host factors associated with the patients. The host factors considered are here. I will come again later for these factors. From the patients, venous blood samples were collected to be used in the antibody assays. In-house established different types of enzyme-linked immunosorbent assays. So ELISAs were used to assess anti-PVDBP, IgM, IgG, G1 to G4 antibody levels, and functionally specialized binding inhibitory antibodies. We used 2 recombinant proteins for these ELISA assays. One recombinant protein is the recombinant protein PVR2, which is representing the critical binding region of the native Plasmodium vivax Duffy binding protein. And the second other recombinant protein is known as NDARC-IG, representing the Duffy antigen receptor for the chemokine on human RBC for these ELISA assays. First, we use indirect ELISA assays to measure the prevalence and magnitude of 4 these different types of antibodies. So we are basically concentrated on the prevalence and magnitude of these antibodies. So this is a schematic diagram that shows the basic steps of the indirect ELISA, where I am not going to describe detail, but in brief, here we are quoted recombinant protein PVR2, and then we added patient's sera. If patient's sera contain anti-DBP antibodies, they will bind to the recombinant protein, and after completing with this assay, a specific color will be developed, and this based on the intensity of this developed color, we can measure the magnitude of the antibodies inside the patient's sera. And second one is ELISA-based binding inhibition assay to measure the functionally specialized anti-DBP binding inhibition antibody responses. Here, we quoted the Duffy antigen, the recombinant protein that represented the Duffy antigen on human RBC, and separately, in a separate container, we pre-incubate PVR to the parasite Duffy binding protein with